bring this over all?
No, I want it to be always the same time every year, and this is totally trumped up.
Okay, and why the same time every year?
Well, just I'm trying to figure out what's going to happen, so you don't want if things are at different times and things aren't synchronized, then you'd have to treat it differently.
Okay.
It's not. I mean, it's nothing specific about those seasons. It's just that you want everything to be happening in sequence, so it's much harder to figure out what's going to happen.
Okay, I mean, for the sake of like the example, like altruism, like him.
Well, no, in reality, of course, these things go on in all sorts of asynchronous ways. But in fact, usually when people do this kind of modeling, they do things which don't seem fundamentally to matter very much, that make the calculations much simpler. And that's what's going on here. Let's see. So first of all, before I start, I should mention that. So you've supposedly all now finished with the quiz. Excuse me, the study questions. The CCLE calls it a quiz. So I just posted the answers and explanations a few minutes ago, so you can look at those. You should look at those. Those would be very important to make sure that you're even if you got them all right. I would recommend reading them over because you might have gotten some of them wrong for the wrong reasons. And so that would be helpful for you. Again, let me remind you, this is for all the study questions that I asked, some of the questions are a lot harder than anything I would ask on the exam. Not all. Some hard questions on the exam. But if you think something's terribly hard, don't panic. It's probably that hard on the exam. On the end exam. Okay, so we have a little bit more about evolution to talk about this morning, and then we'll switch to our next topic. So we talked about natural selection, and we pointed out that when applied in a straightforward way, it tends to create traits which further the self-interest of the individual and the individual's immediate progeny, but not to be very forward-looking, and certainly, according to the theory of natural selection in its bald form, you wouldn't expect evolution of traits that would do things that would help somebody else in a group, but be a disadvantage, cause a diminution of the reproductive success of the individual doing the thing. We call those traits altruistic. That's the biological definition of altruism. So it appears that we and also some other animals have such traits, so the question is how they could have evolved. And it's been a difficult problem for evolutionary biologists to some extent. And there are a variety of different ideas as to what could have led to the evolution of such traits. And Dawkins talks about several. We're talking about a couple of them here. We're going to talk about two of them, basically. First of them is what's often referred to as kin selection, and that's what I introduced you to at the end of the hour last time. The idea essentially is that, so in this example, we've got these, skooks, there it is, yeah. The species of skooks, which are these little guys, and the skooks have a predator, the green guy who doesn't have a name, and skooks typically have a fairly large brood, and then when they've gotten big and plump, once per year, the predators take go and attack them, and the usual situation is just one attack per family per year, simplified to make it easy to do the calculations. Typically, under ordinary circumstances, the predator gets 6 out of the 8 in a clutch, in a litter, in a brood, and 2 of them get away. But there are some individuals who have an allele on some gene that makes them altruistic. Again, this oversimplification that we're talking about a one gene thing. In real life, this would never be a matter of one gene. But we do the calculations. As Dawkins does, we're assuming that. So we're assuming, in fact, for starters, that there are 0.5% of alleles of the alleles in the population are the heroic type, that is, say, the altruistic type, versus the non-heroic, non-altruistic type. So if the form that heroism takes in this particular species, in the skooks, is that heroes, when they see one of these predators, make a loud squawk, which basically does 2 things. It warns the rest of their family so they get away and don't get eaten up, but it guarantees that they will get eaten up. So it's obviously a highly altruistic act, because they don't have any progeny at all. And so the question is, probably from the way I set this up, it's pretty clear that under the right circumstances that this altruistic trait would increase from one generation to the next to some extent, but I actually want to do the genetic calculation. In the version of the slides that you have, I put it all on one or two slides, and it's sort of daunting that way, and so I've split it up now. And I like it better. You will probably like it better too. And so this is the version that you'll see when you look at the recorded thing, or if you remind me to when I send the revised slides. Okay, so we're going to make the calculation several steps. First of all, what we know is that we're starting out with 0.5% of the alleles in the population, being the altruistic as opposed to the non-altruistic type, being the hero type. So assuming that, that's what it says there, 0.1. Then, the first question is, we want to calculate the overall kinds of genotypes in the population. Big H, little h, big H, big H. Remember, everybody has 2 chromosomes, and there's going to be maybe one allele on one chromosome and possibly a different allele on the other chromosome. So I want to calculate that. I also then want to calculate the genotypes of mating pairs. So in order for the offspring to be produced, 2 individuals have to come together and mate, and depending upon which 2 individuals come together and what their genetic composition is, that will influence the composition of their offspring. So I want to calculate the probabilities of various genotypes of mates. Then, having done that, I'm going to calculate the genotype of the offspring, the probability of each possible genotype of the offspring, and then I'm going to ask how that changes after an attack. And then finally, at the end, we can tally it all up and figure out what percentage of the hero gene there now is. So we started out with what percentage of the hero gene there now is, and see if it increased as a result of this process. Okay, first of all, what are the genotypes in the population? Well, there are various possibilities. One is that there's that both on a given individual, both chromosomes have the non-heroic allele. One possibility is that both chromosomes have the heroic allele, and then there are 2 different ways that there can be one of each. Either the one that came from the mother and the one that came from the father, or one that came from the father and one that came from the mother. Yes.
Are these dominant traits, or is it
These are assumed to be dominant traits. Yeah.
Okay.
Yeah. For the sake of the example, the dominant traits. Yeah. That is, even if you only have this gene on one of the 2 chromosomes, it gets expressed. It affects the behavior. Yeah.
Will we cover how this could be different if it was just
no? You're welcome to try to figure it out for yourself. But in actual fact, when the allele is as rare as we're assuming here, there would be almost nobody that would be homozygous for it.
Okay.
So it wouldn't be expressed ever. Yeah. It wouldn't have any effect on behavior at all, hardly. I talk a little bit about what causes things to change from recessive to become recessive or dominant in that thing that I dictated that video on. Okay, so first of all, what is the probability of a little h, little h? Well, it's the probability of if the percentage is 0.5%, then the probability is going to be 0.005 because you divide by 100 to get from the percentage to the probability. And so the probability of 2 little h's is the probability that on the one that it's a little h times the probability that on the other it's a little h, which is this times this, which is pretty high. It's 99%. The probability of heterozygosity can occur in either of 2 ways. It can be either this way or that way. The probability of one way is that, and the probability of the other way is that, and either of those is heterozygous. So the probability of getting some kind of heterozygote, some kind where there's just one but not the other gene is the hero gene, is 0.01, and that's 1% of the population. So we have approximately 99% are homozygous for the non-heroic trait. Approximately 1% are heterozygous for the heroic trait or the other trait. And if you do this calculation, the numbers are so small that you might as well forget about them. So there's virtually nobody that's homozygous for the heroic trait, given that you started out with such a small percentage in the first place. Sorry, which one?
For, P equals prop H, that's the top one. Is that supposed to be the top page?
No, yeah, that's a mistake. Oh, wait.
And then for the
Yeah, no, that's a mistake. Yeah, I said that wrong. It should be little h, little h. Yeah.
And then what about 1 minus point?
No, so the probability of a little h is 1 minus the probability of a big H.
Okay.
So that part's right, but you're right. Thank you. That's wrong. That should be little h up there. I'll try to remember to change. If you send me an email reminding me to change it, I would appreciate it. Okay. Now, pair formation. So we got 99% have one copy of the gene, and sorry, 1% have one copy of the gene, and 99% have no copies of the hero gene. Now, individuals mate, and there are various possibilities and various combinations. You can go through these yourself. I'm not going to work through it in detail, but it's also spelled out there. The bottom line of this is that 98% of the mating pairs have somebody with an H, a big H. And 2%, sorry, I said that backwards. 98% have no hero gene. Neither parent had any hero gene. And 2%, at least one parent did. One or both have parents had hero gene. Okay, so that's easy enough. So these are the numbers that we need to take away with this, and you can look at the details of that slide on yourself. And that's basically what I just said down there in that box. Okay, so let's look at what happens per 100 matings to make it easy. So that was basically kind of calculating the percent. So per 100 matings, if there are 100 matings, on average, you'll have 2 pairs that look like this, where you... Oh, right. Sorry, let me go back there. Sorry, I do want to go back. Let's go back to previous. Whoops. Come on. Slide a minute. Yeah, I didn't say something I should have said. Okay, let's go over that box, because I should have said something that I didn't say that's in there. So, for 98% of the pairs, both parents are homozygous for the non-hero allele. For 2% of the pairs, one parent is heterozygous for the hero allele, and one does not have it at all. Virtually no pairs have both parents having the allele, because the probabilities are so low. I mean, there's some probability of it, but it's negligible. It's 0.01%, so you can forget about it. So basically, you're never going to, you're hardly ever going to have both parents having the allele. So typically, you're going to either have none or one. Okay, so you're going to have 2 pairs in which one parent, out of 100 matings, you're going to have 2 pairs in which one of the parents is a hero, or has a hero allele. And you're going to have 98 pairs in which nobody has a hero allele. They breed and produce, each breeding pair produces a clutch or a litter of 8 kids, 8 offspring. And what's the genotype? What is their genetic composition going to be? Well, since one parent has one, half of them, because of independent assortment, this is what we did in that video talk, and you all probably know this from high school biology or wherever. Anyway, half the kids on the average are going to have a hero allele, and half don't from these pairs. So there'll be two litters looking like this. And then, of course, if neither parent had it, none of the offspring are going to have it. And that's 98 of the pairings. So 98 litters are going to be total non-heroes. So that's the picture. Okay. All right, so that's our background. That's before the predator attacks. What happens when the predator attacks? When the predator attacks, over here, all these types, all 98 of these, all but 2 get wiped out, because that's what the predator does. Over here, somebody squawks. Whoever who squawks first is the one that counts, and the other heroes get away as well. So whoever squawked first gets clobbered, gets killed, gets eaten up, but all the rest of the family survives. Okay, so we end up, over here what's surviving is, we have 2 litters with on average 3 altruists and 4 non-altruists. And over here we have 98 litters with 2 non-altruists. We total it up, we have over here 6 altruists and 8 non-altruists, and 100, and over here we have 196 non-altruists. Put it all together, and the proportion of individuals having an H gene is ends up being 3%. You can look at that in detail at home. So it went up. We started out with 1%, and we ended up with 3%. So it goes up. All right. Now, a question that somebody asked me last time, which I didn't know the answer to, was how far will it go? So it goes up, but does it keep going up and up and up till it's 100%, or does it stop short of that? And so I answered him by actually doing the calculations. So I know the exact answer, but I would like you to try to answer that question just by thinking about it. Do you think it would go all the way to 100%? So basically 3 possibilities. One possibility is that eventually on the long haul things will go back down to very close to 0, or it just won't get very far in the first place. One possibility is it'll go to almost 100%, almost everybody will be carrying the hero allele. And one possibility is somewhere very much in between. And that's actually a question that is on the quiz. Hard question. I might end up writing it, marking it right no matter what you move as an answer. But I want you to think about it because it's good for you.
Something you won't know until you get the answer to that. If we do it and we get it wrong, we won't know we actually got it wrong.
Actually, you will know if you get it wrong. You're told for each. Actually, you will. You're told for each question whether you're right or wrong. You're just not sure what the right answer is. At least that's what's supposed to be happening. Isn't that what's happening? Yeah.
Well, I think you mark it right regardless.
Well, no, I might actually. As I think about, I said I might, but now I'm realizing I've already just posted the answer sheet this morning, so I didn't. I didn't mark. You'll be able to tell what the right answer is by doing it a few times, even if you guess. But the important thing is to understand the logic of it, because that's what's going to help you for exams later. Okay. Now this kin selection idea, this is a trumped up example of try to make it crystal clear, and to actually work out the genetics for you. This happens in real life, but not real often. It happens in real life. There's some animals, and maybe some human groups during evolution, but certainly some animals that have a social family, have a social family organization such that they live together in large family groups. I mean, you have that kind of situation. This kin selection thing can work in real life. And so, for example, there are, in fact, animals that have, exactly like our scoots, that have alarm calls that attract predators away from the rest of the group and allow them to get away and get the squawker gets not. That really happens in real life. And you have another example is you have you have cases of animals where some animals carry a gene that makes them both terrible tasting, or even poisonous, and makes them brightly colored. So predators go after them and take one bite and say, "Oh God, I don't want to eat this species anymore." But it's obviously really bad for them because they're very noticeable. And a lot of that, both those kinds of things occur in nature. Now, so it's interesting that this provides a mechanism for kin selection, or a mechanism for the evolution of altruism under certain limited circumstances. But what it also is good for, this example, is that it makes it very clear. If you can have a trait that is terrible for the person that has it, but it's good for their genes, then, that's the essence of what natural selection is all about. And some wag at the time when these things were first, when Darwin wrote, said, if Darwin were right, a hen would be nothing but an egg's way of making another egg. That's exactly a good characterization of the way it is. We'll talk a little bit more about that in a few minutes. I'm taking way too long with this, but that's all right. That's not all right, but it's going to happen. Now, this kin selection idea is very specialized. Another idea that put forward was actually put forward by Darwin himself first, and then by some other people later. Darwin said, in The Descent of Man, groups with a greater number, he didn't use the word groups, but anyway, groups with a greater number of courageous and faithful members, that's altruists, because they're courageous. They'll sacrifice themselves for the group. Courageous and faithful members who are always ready to warn each other of danger and aid and defend each other would spread and be victorious over other tribes. And he thought that might be, he used that as a possible explanation of how it could be that these group advantageous but individually disadvantageous traits could spread. A later worker, a guy named Wynne Edwards, who I believe was talked to a little bit about, mentioned a little bit in Dawkins, in the part you've read, argued that that if you have traits that favor, that you can have traits favoring the group evolved in a more subtle way. The animals don't actually have to battle with one another. The idea was, for example, you might have a trait of self-sacrifice of some sort, which helped the group as a whole survive in lean times, after droughts and things when there wasn't much food or whatever. And other groups that didn't have those traits would just die out, leaving the ecological niche that they had lived in available for this other tribe that barely managed to survive to repopulate. And so that would be a way that would happen rapidly once the first tribe was gone, and when lean years were over. And so that would allow for a rapid expansion of those groups that had these altruistic traits. Here is so the problem, however, with this sort of with both Darwin's idea and Wynne Edwards' idea, and the biologists have criticized it greatly, and nobody believed it at all for many years until fairly recently, is that there's also going to be whereas it's true that these groups that have a lot of altruists have certain advantages over groups that don't, and may sometimes be able to take over their territory and thus expand. It's also the case that these traits are personally disadvantageous. And so you've got something promoting the evolution of the trait and something working against the evolution of the trait. And it's been until recently it was argued that the within group bad things were going to be more potent than the between group good things. And so in fact these traits, group selection would never work, it was thought. This is an example of what would happen. So suppose you've got the red guys are the altruists, the black guys are the non-altruists. These gray arrows mean that under whatever circumstances that this group will probably end up taking over the nearby group. In each case, it's the one with the greater number of altruists taking over the group with a lesser number of altruists. So I just put these are just sort of some possible things that might happen. If that did, and right now we're starting out here with 19 red. If you take the whole population that's on the board, we've got 19 red guys and 189 guys altogether. That's the full population. So 19 out of 189. The 189 stays constant. The assumption in these kinds of calculations is that the group always breeds up to the maximum carrying capacity of the environment. Okay, but now suppose we have this slow within-group decline of the trait, of loss of the gene, but due to these gray arrows, we have some expansion as well. In this example, what happens is, you notice it goes up from here to here, and this group, because what happens is these guys that had more of it went out and repopulated, but this number here is not as high as that number was because there's also this attrition due to within-group decline of the trait. Again, this group, actually this group is the balance. This group, nobody had it here, one guy's got it there. Put it all together, and we went from 19 out of 189 to 15 out of 189. So the trait wouldn't really grow, and that's kind of because of this problem that within group the decline being greater than the advantage between groups. But relatively recently, there's an anthropologist by the name of Bowles who's done a lot of studies, and he says these are bad assumptions about what social structure and life was like at the time that human evolution occurred. He says there's lots of I don't know if everybody agrees with me, and I'm not enough of an anthropologist to know whether this is whether most physical anthropologists would agree. But he says there's evidence that humans at the period when these traits would have been evolving were real. They were really mean, vicious individuals to other groups, and that they would exploit, they would attack anybody they thought they could win against, and wipe out the other group and take over. So this is what we just saw. It's just a recap of the same thing. But here's the picture that Bowles presents. You've got this most heroic group, the group with the most altruists in it, looking around for who it can beat most easily and going after those groups and wiping them out. And now you've got a much different picture. You again do have within-group decline of the altruistic trait, but you've got this colonization by this group that has a relatively large number of individuals with these heroic alleles taking over everywhere. And now it goes from 19 out of 189 to 35 out of 189, almost a doubling in this example, this trumped-up example. So that he argues this process might well have occurred, and that he argues that whereas altruism by itself would not have evolved in human populations, that what would have evolved is sort of there could have been a co-evolution of in-group altruism and out-group belligerence. Now he argues, and you can make your own judgments, as to whether that's kind of how us humans are. He argues that we are. He says we're nice to people like us, and we're not nice to people that aren't like us. And once again, you can decide for yourself whether that's valid. But insofar as it is valid, he says that may be how both the altruistic traits and the belligerence co-evolved and became part of the human psyche. Okay, yeah, these are ideas. They're not facts. This is all based upon computer modeling, but interesting idea, which might have some validity to it. And he argues that there is, in his view, there is anthropological evidence, physical anthropology evidence that supports the idea. I'm sure there are people that argue against it. Okay, so the take-home message here, or messages are as follows. First of all, our traits are the result of an evolutionary process in which traits that increased reproductive success were selected. Period. That's what evolution is all about, according to this thinking. Point one. We can possibly learn things about ourselves by looking at the consequences of the above, such as the fact that we can learn things about ourselves by this. Dawkins talks about a number of things. He talks, for example, about mating behavior and the difference between males and females and a variety of things, and you've read about that. We've not talked about that in class. Second, in its simplest form, natural selection predicts that we will tend to do things that maximize our own and our children's reproductive success without regard to future generations and without regard to others. That was the first example we saw. Point three, natural selection does not necessarily lead to traits that are optimal. Sometimes it does, but under certain circumstances it does not. So that's a fallacy. People often say that it does, but it doesn't. Only sometimes. And fourthly, natural selection suggests that some of our presumably finer qualities, such as altruism towards others, may have evolved only in certain contexts and situations and might have come along with considerable baggage, considerable negative baggage. Now, this is all very theoretical and hypothetical, right? But what it says is it's worth thinking about the possible forces that were driving our evolution in the process of trying to think what we as humans are like. Yeah?
When you say traits, do you mean like the trait makeup of some sort of population?
I mean the trait like altruism, the characteristic.
Okay.
Okay, I think that's... this again is a detailed slide reviewing which I used last time. You can read it, but it's what I just told you was my preferred version. Okay, now major change of topic, new topic. We're going to spend about a week now introducing the nervous system in preparation for some topics in which you need to know something about the nervous system in order to understand it. Most of what we're going to be presenting is kind of background that in its own right, I don't want to say it's not interesting because I think it is interesting, but you may not think it's interesting, maybe some of you. But it has, unless you're a biologist or a neurobiologist, you might say, who cares what a nerve cell looks like, and so on and so forth. You certainly don't care what the various parts are called. But we're going to have to know some of that in order to progress. However, there will be, in this part down here, actually this is not a complete, this outline is not complete, but anyway, in the last part of, this is the first part of what we're going to be talking about, where we talk about the functions of the major parts of the nervous system. There are some things that are of intrinsic interest. So for the most part, for the next week, the stuff won't be intrinsically interesting. I hope the stuff so far, some of it's been intrinsically interesting. And this next block of material you may not like as well, but it's what we need to go on, and there'll be some parts that are interesting. Okay, first of all, the nervous system is made up of nerve cells. And nerve cells are very different from all other cells. Most cells are kind of, gloss. I mean, they may be elongated, they may be flattened, and so on and so forth, but they're basically gloss. Nervous systems are very difficult. They have all these tentacles sticking out from them. And here is a picture of a, in quotes, typical nerve cell. It's got a part that's like other nerve cells, that's like other cells, called the cell body, or the soma of the cell. And by the way, you'll have to know all these words now. Soma or cell body. It's got a nucleus in the middle with the genetic material. So that's like all cells. But then it's got these tentacles sticking out. It's got two kinds of tentacles. On the one side, it's got what are usually long and often very branching tentacles that go a long ways away from the cell. It turns out that what nerve cells do is they communicate with one another. They're communication devices, and they also do other things. One of the things they do is they communicate. They send messages. They send their messages along these long tentacles, these long fibers. And these messages are called nerve impulses. There are a bunch of other names for it. We'll talk about that later on. But I'll try to remember to call them nerve impulses. Or maybe sometimes nerve messages. And they typically start here, if they're going to start, and run along to the end of these tentacles. If they come to a branch point, they go into both branches. These long tentacles are called axons. So they typically have these long branching axons. Now, at the end of each branch, there is a terminal swelling, sometimes called an axon terminal, or some books, unfortunately I don't think yours, although actually I'm not sure about that, called terminal buttons, or more properly they're called boutons terminaux. That was the name that was originally given them by the anatomists that described them, but modern textbooks find that too chichi to use French, so they translated it to terminal button, a terrible term, I hate it. I will hopefully never use it. Axon terminals is the term I prefer. These axon terminals are crucial in one nerve cell communicating with another. We'll talk more later about the details, but these terminal buttons secrete chemicals. And these chemicals affect other nerve cells. So at the other end, let's look at the other end. At the other end of this nerve cell, we have some more tentacles, but they're not, and they're often branching a lot, but they're never long things. They're usually not long things like this. They may branch a lot, and we'll see some pictures, real pictures in a moment, but they're typically more bushy or tree-like rather than being long things with an occasional branch. And the terminals from one nerve cell connect up with, or make contact rather, with these branches at this end of the cell. So this is the output end of the cell. These nerve impulses will start and go out here. This is the input end of the nerve cell, where information comes into the nerve cell from other nerve cells for the most part. So when the axon terminal of one nerve cell makes contact, where it makes contact with these tentacles here, we call that a synapse. What's that term? It's out here somewhere. Synapse. That's the connection of one. That's the place where one nerve cell contacts another nerve cell for the purpose of communication. We'll talk more about exactly how they work and so on later, in a few lectures down the line. These tentacles here are referred to collectively as dendrites. So we have axons and dendrites, the output end and the input end. There also are two kinds of input synapses. There's excitatory ones and inhibitory ones. We'll talk about that later. Don't worry about that for a moment. Okay, let's see. Now, these synapses always have an input side and then a target. We refer to the input side as the presynaptic side of the synapse, and the target side, the other side of the dendrite, the dendrite itself, the part which is receiving input from another nerve cell, as the postsynaptic side of the synapse. It's often the case that, almost always the case, that any given nerve cell gets input from a lot of other different nerve cells. That's what's shown happening here. Each of these branches is probably from a different nerve cell, or many of these branches from different nerve cells. We therefore say that many nerve cells converge their input onto various target nerve cells. That's the term of convergence. Whereas at this end, any given nerve cell provides information to lots of other nerve cells. It synapses on lots of other nerve cells, and that's the concept of divergence. Nerve cells make messages, nerve impulses, which are, in quotes, neural messages. Nobody but me calls them neural messages. Everybody else calls them nerve impulses, and there's also some other official terms. What these messages are is an electrochemical disturbance that we'll talk about later on that travels along the axon at about, it's for different nerve cells, depending upon their exact structure, it may travel anywhere between 1 and 100 miles an hour. So it's kind of the kinds of range speeds that cars drive. Pretty fast given the short distances involved, but not like the speed of light or the speed of sound or something like that. Just quite fast, but not terribly, terribly, terribly fast. These messages are all or none. There are no big messages or little messages. They either there is a message or there isn't a message. It's like ringing the doorbell. Either somebody did it or didn't do it. Oh, and I guess I did want to mention, so there's excitation, the fact that nerve cells can encourage other nerve cells to fire, and there's inhibition, the fact, which I've indicated with the blue guys, that some nerve cells, input from some nerve cells can discourage the target nerve cell from becoming active or producing a nerve impulse. And we'll talk more about that later on. Yes?
Those are not really connected to anything else, but they are.
So these guys, yeah, these came from other nerve cells.
And the bluish purplish circles.
And they all came from other nerve cells. Yeah. I should have drawn them off the edge of the slide. Good challenge. Could change that. Okay, now I'm going to talk in a few minutes. We'll start talking in a few minutes about the specific anatomy of the mammalian or the human brain. But I want to talk about some general aspects of all nervous systems first. So here's a generic nervous system. There's the border of the nervous system. Inside the nervous system, you have nerve cells, cell bodies, axons, and dendrites. In many nervous systems, certainly in our own, what happens is the cell bodies and their dendrites are clumped together close to one another in regions referred to as nuclei. That's not the nucleus of the cell. This is a different use of the word nuclei. So a nucleus in a nervous system is a region where there are a lot of cell bodies and dendrites all, close to each other. And of course, there's some axons too, but the conglomerate, the cell body, some of the axons may travel distances within the nucleus and may also leave the nucleus. You typically have many different nuclei within a nervous system. I'm just drawing 3 here, but in us there are thousands, hundreds and thousands. The axons of one nucleus may go may go to some other nucleus along axons that go from one nucleus to the other. Often these are bundled together. When they're bundled together like that, they're referred to as tracts. Often these tracts have a fatty insulating substance on them that makes the nerve impulses go faster, and that is called myelin. The tracts are said to be, nerve fibers are myelinated. Each fiber is wrapped. Each nerve fiber, axon, is wrapped with a fatty coating that makes the nerve messages go faster. Because the fat looks white, because they're all bundled together, if you actually look at it, you slice it and look at it, it looks white, so it's referred to as white matter. Whereas if you look at the cross-section of one of these nuclei, it will look gray. So they are gray matter, typically. So we talk about white matter and gray matter. Gray matter is where the synapses are and a lot of things going on. White matter is where you're having information being transferred from one place to another. Nerve messages, nerve impulses going from one place to another, from one nucleus to another nucleus. Sometimes tracts of axons leave or enter the nervous system. If they're outside the nervous system, they're not referred to as tracts anymore. They're referred to as nerves, but they're basically the same thing. Finally, almost finally, not quite finally, there are three categories of nerve cells in the nervous system. There are some nerve cells that carry information that carry information into the nervous system. They are called sensory neurons. There are some nerve cells whose cell bodies are in the nervous system, and they carry information out of the nervous system to muscles and things like that. They are called motor neurons. And then there are lots and lots and lots and lots of neurons that don't either, that basically just communicate entirely within the nervous system. They get their input from they get the things that affect them, information that affects them from other nerve cells, not from the periphery, and they influence other nerve cells, not muscles or glands. They are all collectively called interneurons. So 99% of the neurons in your nervous system, the nerve cells in your nervous system, are interneurons. And a very small percentage of them are sensory neurons or motor neurons. Finally, in a real nervous system, you may have a number of things like this with nuclei in them, and they are sometimes called ganglia, which is a term I don't have on here. But you typically have a congregation in most animals, certainly in all vertebrates, and in most invertebrates. You have a congregation of nerve tissue up at the front end, at the end that is that has the distance receptors on it, like eyes, ears, and noses. That is the end that gets that encounters information from the outside as the animal progresses. And it walks along, or swims along, or whatever that flies along. And those frontward parts of the nervous system get much more elaborate than the rest. Huge amount of information processing is done in them. And they are, by definition, what we call a brain. So a brain is simply a front-end congregation of neural tissue. So I have a good friend, a colleague, who a couple years ago wrote a book called Brain Bugs. Anybody read Brain Bugs? Anyway, and he asked me to read some chapters, and he commented that worms don't have brains. Well, that was absolutely wrong, and he knew it was wrong. I mean, I told him he should be ashamed of himself, and he changed it and apologized. He apologized to me in particular because I happen to work on these kinds of animals that he said don't have, I know, not worms, but other animals that he would that don't have brains. Anyway, so brains are the frontward congregation of neural tissue, and almost all, jellyfish don't have them, but most animals have them. And these are not only, there's a lot of information processing done there, and not only are they bigger and fancier than the rest of the nervous system, they typically end up taking over and kind of controlling the rest. They become dominant. Okay. Now, actually, let me just go back to the slides for a minute. Another important thing about all nervous systems, and certainly about advanced nervous systems like those of the mammals, and certainly those of ourselves, are that these different nuclei are commonly specialized for doing different information processing tasks. They're not all doing the same thing, and this is referred to as modularity. And, Professor Zeidel, during his class of half of the class, will talk about that a great deal, and we'll actually talk about it quite a bit in a little while too. But so different parts of the brain, different nuclei of the brain, have different jobs. And if you simply look at them anatomically, you can see very, very different structures. If you look through a microscope at the nerve fibers, at the nerve cells. So these are cross sections of the brain in three different parts of the brain. Any neuroscientist can look at those and tell you where those, which region of the brain that is. They're obviously look very different from one another, and that reflects the fact that they're doing different jobs. Let's see, is there any area we're going to be talking about a lot more later than example one here? Yeah, that's. Well, this is a part of the brain called the hippocampus, which we'll be talking about a certain amount at the end of my lectures. And this is visual cortex, which we'll be talking about it's the next block of lectures. This is cerebellum, which I don't think we will be talking about, but the cerebellum's got these amazing nerve cells. Here's the cell body, there's the axon of the sky, and all this branching stuff is the dendrites of these two nerve cells just those two nerve cells right there. Huge branching. That's exceptional. They're so extreme, but it's so neat that I wanted to show you some pictures from real pictures. They were first described in detail by a Spaniard named Ramon y Cajal, very, very famous. Here is a modern picture where somebody injected a dye into the cell body, and the dye spread all over it. So you can see these guys. Huge branching trees from just one nerve cell. This is the cannula that's injecting the dye. And they're laid out in a very organized fashion in the part of the brain called the cerebellum. And this is all, I'm just showing you this, you don't remember. Okay. So those are general aspects of the nervous system. Modularity, tract, nuclei, white matter, gray matter, so on and so forth. Motor neurons, sensory neurons, interneurons. Let's now talk a little bit more specifically about nervous systems. And to start with, we need a framework for geography, sort of like latitude and longitude. And the framework that's used is shown here. The framework is developed based upon animals, four-legged animals, or not also worms, but things that don't stand on 2 feet like us. And so in such animals, the top side is dorsal, the bottom side is ventral. Dorsal, ventral. The front end is rostral or anterior. These are all terms you need to know. The back side is caudal, or backward direction is caudal or posterior. There's a midline. Most animals are bilaterally symmetrical, looking the same on the left and the right. And we refer to moving away from the midline as moving in a lateral direction. Lateral. Moving toward the midline referred to as medial. In us, it gets a little bit more complicated. So if I'm standing here, my head is oriented the same way it would be in an animal. It's upright. So my head, it's dorsal, ventral, lateral, anterior, posterior. That's fine. But my body is. If you want to figure out how to name the part, how to use this nomenclature for the parts of the human body, the easiest way to do it is just to go down like this, and then it'll be the same as an animal. If you don't do that, it's kind of screwy. So front becomes ventral, back becomes dorsal. It's kind of screwy. But if you think of yourself for a moment as a four-legged animal, then it becomes very easy to figure out. So if you take a test, if I see you all falling on the floor, I'll know why. Okay, so that's some nomenclature that you need to know. Now, here is the nervous system, or at least the top part of the nervous system, and human. And I have labeled certain things. First of all, the brain is here and is attached to the spinal cord. Okay, I've shown this. The spinal cord down here, which I've shown in pink, has also some other pink stuff. It runs around like here and up to here and around to here, like so. I mean, you can't see it's pink, so there should be a slightly pink cast to this. It's referred to, the parts up here that are pink, above the spinal cord itself, are called the brainstem. Term you need to know. So it's spinal cord and the brain, and the bottom part of the brain, or actually the caudal part of the brain, is called the brainstem, and we'll talk about what's in it in a while. Then, covering the whole thing is what's called the cerebral cortex, and I've shown that in blue. And then, if this is the spinal cord, and this is the rest of the brainstem, this part here with my fist on it, and there's this covering, which is the cerebral cortex, sandwiched in between the two. If this is lateral here, so dorsal, ventral, there is a big glob of tissue there, a big glob of tissue here, and then there's this cortical sheet covering it all. These globs that are underneath the cortical sheet, but outside the brainstem, are called the, actually I have to give you a word for them, but they're there. I'll give you a word in a little while. But the combination of these, they're the green parts. The green and the blue together are called the telencephalon. So there are basically 3 parts of the brain here. Blue part, the pink part, and the green part. The green and the blue are the telencephalon, spinal cord, brainstem. That's everything that's that slide. Cerebral cortex.
The cerebral cortex is just all
The blue stuff.
wiggly
All the blue stuff.
I'm just trying to think of like in the head. It's just like everything that isn't those two things.
Yeah.
Okay.
Okay, now let's look first at the brainstem. Yeah. Where are you? Question? Yeah. Yes. It's one of the two parts of the concept. Well, 2.5. It's one of the parts of the concept. Yes. Okay, brainstem. Brainstem. Okay, here's the spinal cord. Brain starts here. You can it's marked by where it starts by swelling here. We'll tell you what that's called in a few minutes. But let's just look at the spinal cord itself for a minute. If we take a cross section through the spinal cord anywhere along its length, it runs all the way down from about there to right there. It'll look about the same. And this is what it looks like in cross section. Let's look at that in a little more detail, except not today. Goodbye.